We'll continue our discussion of development, fluctuation, and adaptation of populations by discussing fluctuation. Our objectives are to identify fluctuations in concentration and composition of microbes that occur after feeding and feeding forage versus concentrate. As for the last lecture, there is no reading from the textbook. Bacterial concentrations reach a nadir after feeding. We can see that in the figure to the right. This result may be a bit counterintuitive. You may think that with the introduction of feed and new substrate, that bacterial concentrations would increase immediately. There are several explanations for why we may still see the nadir. First, there may be simple dilution with feed. There may also be introduction of oxygen with feed, which could kill bacteria and be responsible for the decrease. Feeding increases passage rate, and it's possible that passage of feed with attached bacteria is also responsible for the decrease in concentration. Concentrations of bacteria remain low, but they slowly rise in this case about 8 hours after feeding. At about 20 hours after feeding, they're at their peak, and then they slowly decrease until the next feeding. Keep in mind that in this and most of the other examples we'll see, the animals were fed once a day, and that was for proof of concept. That does not reflect practical feeding conditions. In the last slide, we looked at direct counts of bacteria. We look at culture counts, as does the figure on the right. We can see the same general pattern as direct counts, but the pattern is more exaggerated. This is consistent with the idea that oxygen enters the rumen and kills bacteria. It may explain why we can culture relatively few after feeding. For methanogens fed a high forage diet, it was found that concentrations reach a nadir several hours after feeding, and then they slowly rise to pre-feeding concentrations, much the same as with bacteria. With a high-grain diet, the pattern was not as clear. This kind of experiment has never been replicated. For the entidiniamorphid protozoa, the concentrations reach a nadir after feeding, slowly rise, and then gradually decrease until the next feeding, just as for bacteria. The growth rate is shown here, and it shows the opposite pattern of concentration, which is what we would expect from the data with bacteria as well. For isotrichid protozoa, the concentration follows a bit more of a curious pattern. The apparent concentrations reach a nadir long after feeding, and then they seem to rise precipitously before the next feeding. We can see that in the figure at the right, both for daisy-trichs and isotrichs. Further, the concentrations continue to rise even if additional feed is not given. This pattern is really hard to explain by itself. The explanation comes from the observation that isotrichid protozoa are found on the ventral wall after feeding. We can see that in the figure at the right. The figure at top shows the rumen wall with an arrow pointing towards microbial biomass. It's the whitish material. The figure below shows that microbial mass under the microscope. You can see the cells packed in, and they're mostly isotrichous species. The probable scenario is that isotrichids engorge themselves after feeding and store carbohydrate. They then settle on the rumen wall, and they cannot be detected in normal counts, like in the figure on the last slide. They rise again in response to feed or depleted carbohydrate stores. In the last slide, when they weren't fed, they were apparently increasing in concentration just because they are rising off the ventral rumen wall because of depleted carbohydrate stores. It's amazing how well synced the protozoa were with the next feeding, as if they were anticipating that they would be fed. We see a similarly curious pattern with zoospore concentrations. These rise precipitously after feeding, and then decrease until the next feeding. The reason for this is that the zoospore is not the only form of fungi. Their fungal reproductive cycle is induced by a dietary component and causes the zoospores to be released from the sporangia. As the sporangia concentrations fall off a cliff, the zoospore concentrations rise precipitously. The dietary component that induces the release of zoospores from sporangia is a heme-containing compound. The figure at the right was not done in vivo, it was done in vitro, and an inducer and oat extract that contains that heme compound was added. To this point, we've discussed changes in concentration that occur after feeding. For the rest of the lecture, we'll talk about changes that occur when feeding a grain versus forage diet. This is the study we saw in the lecture about quantification of microbes. It quantified total and culture counts when feeding a high forage versus a high grain diet. From the table, we can see that feeding the grain diet increased both direct and culture counts. The absolute abundance of all groups was increased, including the cellulytics, those that grew on the cellulose-cellobiose medium. But when we express these values as a percentage of the total culture counts, that on the complete carbohydrate, we see that the percentage decreased when feeding higher grain. The percentage of glucose, starch, and pectin utilizers, by contrast, increased. And a question you can ask yourself is, do these trends make sense? Does it make sense with feeding higher grain? We increase the concentration of total bacteria, but we decrease the percentage of cellulytics. The last slide looked at functional groups of bacteria. The current slide focuses on species. We see when animals were fed a high grain diet, the percentage of Butyrvibrio and Fibrobacter decreased, whereas the percentage of Selenomonas, Ruminobacter Haemophilus, and Lactobacilli increased. You can ask yourself again, do these trends make sense? Does it make sense that with feeding grain, we would decrease Butyrvibrio, Fibrobacter, but increase Selenomonas, Ruminobacter Haemophilus, and Lactobacilli? I'll leave that question to you to answer. The last slide looked at culture counts. The current slide also enumerates species, but using a culture-independent approach, qPCR. We can see that the concentration of several bacteria decreased. Bacteria including Fibrobacter succinogenes, Ruminococcus flavifaciens, Prevotella ruminicola, Eubacteria ruminantium, Treponema bryanti, Streptococcus bovis, and Succinovibrio dextrinisolvens. Prevotella bryanti and Selenomonas ruminantium, by contrast, increased. And I think you can ask yourself again, do these trends make sense? This is a more difficult study to interpret, and so I'll help you through this one. It does make sense that Fibrobacter succinogenes and Ruminococcus flavifaciens would decrease, because they're cellulytics. The other decreases, though, make less sense. For example, Succinovibrio dextrinisolvens uses maltodextrins. We would expect its concentration to increase after feeding grain. The study was carefully performed, and it's difficult to dismiss it simply because it doesn't mesh with our understanding from culture-based studies. It suggests that there's some bias or other deficiency in culture-based studies, and we need more of these qPCR-based studies to understand more about the ecology of the rumen. Protozoal concentrations increase until grain is about 60%, and we can see that in the figure at the right, where protozoa increase substantially after being switched from a 0 to a 60% grain diet. Above 60% grain, concentrations decrease. Even though there's more substrate available to them with the increase in grain, the minimum pH decreases. Isotrichids are especially susceptible to this decrease in pH, and they can disappear entirely. The Entadyniomorphids are more resilient, but on the highest grain diets, they can still disappear. For fungi, the differences are not as clear-cut. First of all, there's a methodological difficulty in measuring fungal concentrations. The data that we do have suggests that a high forage diet, particularly with long particles to increase retention time, generally favors higher populations, but it's hard to make more specific statements than that. For methanogens, in the one study that examined them, there didn't appear to be a large difference in abundance between a high grain and a high forage diet. This is curious because other studies have shown that there's lower activity of methanogens on a high grain diet. There's lower methane production as the percentage of gross energy increases in the diet. This shows that there is not a good connection between the abundance of methanogens and activity. More work is needed. The take-home messages from this lecture are that all microbes reach a nadir after feeding, but at different times and for different reasons. Switching from a high forage to a high grain diet shifts bacterial populations. Those shifts have been monitored by qPCR, and they're unexpected from culture-based studies. The impact of diet on abundance and activity are not fully understood for fungi and methanogens.